Hey guys, I thought I'd make this video to help inform people of what are the major stages in the development of pharmaceuticals. And this was a very high-level topic that I never really got until I was decently into my career in biotech firms. So I hope this is useful and informative to you guys, and to kind of illustrate a very common story that we're going to find time and again. I'm going to be talking about a generic story behind Herceptin. And Herceptin has a generic name of trastuzumab, and the key thing to make note of here is these last two letters here, which tells us that this is an antibody. And the way that pharmaceutical firms like Roche end up making these types of drugs starts with identifying a target. So we're going to ID a target protein. And to give some background into what Herceptin does, Herceptin is used to treat patients with specific types of breast cancer. And what Herceptin does is that there's a protein on the surface of breast cancer cells that is called HER2. And so if you are a woman or a guy and you have breast cancer, you're most likely going to be a woman, but your breast cancer cells are going to be expressing certain proteins on their surface in abnormally high concentrations or numbers. And HER2, specifically HER2 positive cancer cells, so if you turn out to be a patient with cancer cells that are HER2 positive, that means they have lots of HER2 receptors on their surface, you are now a candidate for Herceptin. And so what Genentech did early on, before they even made this drug, is that they identified HER2 as being this receptor on the surface of these cancer cells that they really want to bind something onto, because for whatever science reason, there was some PhD with a background in immunology or oncology that suspected that if we can stick stuff onto HER2, it may reduce the growth rate of these breast cancer cells, and they did a bunch of stuff upstream. But for the sake of this story, the point is that we've identified some target. And in this case, our target is HER2. And that's just a protein that is on the surface of these breast cancer cells. It could be any other protein, and this is why pharmaceutical industry is so big. But in this case, we're going to be sticking with HER2, so we have a nice story to go with here. And what's going to happen after we've identified our target is that we're going to want to start finding out and getting the protein that will stick onto our target molecule. And so you will commonly see things called plasma cell workflows. And in these plasma cell workflows, this is where it pays to have a decent understanding of immunology. What scientists will do is that they will take HER2, or they'll take whatever protein or whatever target you have of interest, and they will essentially dose these mice with lots of your target molecule. And this is to induce the mouse's immune system, its adaptive immune system, to start creating plasma cells that will have a response to the HER2 receptor. And so what's going to happen here is that we inject our target into a mouse, we get lots of plasma cells to make antibodies. So the plasma cells are these B cells that have specialized in creating antibodies that have a high specificity for our targets. So we can think of this stage as engineering the B cells, or we're engineering the mouse's adaptive immune system to target our target by dosing the mouse with this. And this is a process that takes several weeks as you're breeding the mouse and killing the mouse and getting its spleen and getting its lymph nodes and getting the bone marrow to get these plasma cells. And it's a very complex topic that I've touched on in other videos. But the point is, we're going to engineer our B cells, specifically our plasma cells, And then what we're going to do after we've engineered our plasma cells is that we're going to perform plasma cell workflows. And there's a company called Berkeley Lights that has a product called the Beacon. And on the Beacon, we can make use of these microfluidics and optics in order to identify which of these plasma cells is producing antibodies that have a high affinity for a target. And this significantly reduces a lot of the time and money required to identify and export the specific plasma cells that have made the correct antibodies. And what we get out from this process are mouse plasma cells, and specifically mouse antibodies that have a high affinity for our target protein. But what we want to do next is we need to engineer the mouse antibody to be compatible with human antibody. And there's many series of steps here, but we modify the FC region and keep the variable region of our antibody intact. We don't need to get too deep into it for this general introduction, but the point is what we end up getting out of this are the genome. Specifically, we get out the complementary DNA for the antibody, the human antibody, that has affinity for our target protein. And this is done through sequencing. And there's tons of next generation sequencers, NGS machines, that can do this process that we rely heavily on. And you can, common instrument makers are Illumina in this field. And so once we have the complementary or DNA for our antibody, we're going to infect CHO cells. So we're basically going to be engineering CHO cells, Chinese hamster ovary cells, that will produce our antibody. And we're going to do that by, we can use a virus, we can put our cDNA into a virus and infect our CHO cells with that virus, and they'll inject their genetic material into the genome of the CHO cell. And then after we've infected our CHO cells, what we will now need to do is something referred to as cell line development. And in the process of cell line development, what we're doing is we're finding which cells make the most protein, make the most antibodies specifically, divide at good rates, and have very long life cycles, in that they will continue to produce this protein with a low rate of mutations for a long time. And so a lot of stuff goes into proper cell line development, but the gist of this is, and this is another key point where we can use the Berkeley Lights Beacon, is that we can very rapidly get candidates from our CHO cells to see which specific CHO cells do a great job of making our antibodies, and make lots of other cells that will make more antibodies, so you get kind of a snowball effect, and that will also proliferate for long periods of time on the chip, and hopefully downstream in our batch reactors. And so after we do our cell line development, we can move to the downstream processes, which I'll just call this downstream manufacturing. And downstream manufacturing involves basically taking clonal cell lines from our cell line development workflows and sticking them into flasks, and moving up from very small flasks into larger and larger flasks until we get to batch reactors. And by the time we're at the batch scale, typically we're going to have enough of our antibodies that we can sell it in the form of Herceptin or whatever our final product is, so that we can start curing cancer, or at least mitigating some of the bad effects of it for our patients. So that is going to wrap things up for this video. I hope you guys find it useful. Let me know if you have any questions, and thanks for watching.